the first part that we talked about yesterday, chapter 9, I did post yesterday afternoon. I still haven't made a decision about chapter 8. I might try to make a different video for 8, but I haven't had time yet. So I left this up because this is sort of where we left off yesterday, and it's a great place to review a little bit and get started before we move on with the new material. So remember, we're in chapter 9. We're talking about cellular respiration, which is also known as the complete oxidation of glucose. That means the same thing, right? And the first step, remember, you have to know the overall chemical reaction. We said we're going to break this down into 4 steps, and the first step is glycolysis. So I have this on the chalkboard here. I'm going to have to erase it in order to put the rest of it on here because this is all the space I have. But if you have the note outline, you will see a very similar set of sentences, set of pictures in your note outline. I believe what's missing are the ATPs and the NADHs, because if we were doing this in class, I would be expecting you to kind of follow along, right, and fill things out. The last thing, so we have the starting molecule, which is something you have to know, the ending molecules, which in this case are 2 molecules of pyruvate, and we're supposed to keep track of the ATP, carbon dioxide, NADH, and FADH2. And in this case, the only things we have to keep track of are ATP and NADH. So 2 ATP need to be put into this reaction. We gross 4 ATP, so we net 2. And the other outcome is we produce 2 of these NADHs. Remember, NADH, that's that electron carrier. It's carrying hydrogen atoms, and basically what's happening is we're removing these hydrogen atoms in pairs from the glucose 2 at a time, right? So far we've produced 2 NADH, and we're going to need those for later, because this is how we're going to convert this form of potential electrical energy in the NADH form, and it's going to turn into more chemical energy to produce the ATP at the end. Last thing we need to know about each of the 4 steps that I don't have up here yet is where it happens in the cell. And I think I said this out loud, but I know that I didn't write it down. This happens in the cytoplasm. And that is for both prokaryotic cells and eukaryotic cells. And of course, since it's by 1, we're mostly focusing on eukaryotes, so I'm probably just going to stick with eukaryotes for the rest of this. When you come to micro, I'll ask you to know both prokaryote and eukaryote where this happens in the cell. Okay, so that's step 1. Remember, this arrow represents 10 different enzymes, lots of chemical reactions to get us to this pyruvate, right? In a pathway, enzymes in a pathway, each little step, so we only release a little bit of heat, we only make a little bit of entropy so that the cell doesn't boil to death, essentially. All right, so I'm going to erase this, and I'm going to put step 2 up here, and maybe if I have enough space, I'll put it as well. Step 2 is actually called pyruvate oxidation, but we usually refer to it just as called the prep step. And basically what it is it's an intermediate step between glycolysis and the giant Krebs cycle, which also has 6 or 7 or 8 enzymes. This prep step is a single enzymatic reaction, 1 enzyme. Okay? And it's going to pick up the starting molecule for the prep step is going to be what we had as the ending molecules for glycolysis. So I'm going to write 2 pyruvates here. Right? 2 3-carbon molecules, right? 2 pyruvates. That was the ending molecule from glycolysis. It's going to become the starting molecule for the prep step. And we have a single 1 enzyme. And the molecule we produce is something called acetyl coenzyme A. Okay, so we know this word coenzyme from 2 days ago when we were talking about enzymes, this is a helper molecule for the first enzyme of the Krebs cycle or the citric acid cycle. Starting molecule, ending molecule. Now, something you need to know about this ending molecule, an acetyl group only has 2 carbons. Because I have 2 pyruvates, I'm going to do this reaction 2 times, and I'm going to produce 2 acetyl coenzyme A molecules. In your textbook, in any textbook ever, you're only going to see it 1 and 1 because this reaction can be used in the beta oxidation of fats. It can be used to break down amino acids. But in this case, because we're completely oxidizing glucose, we end up with 2 pyruvates from glycolysis. So both of those pyruvates are our substrate for the prep step. Now, because we have 6 carbons over here, but only 4 carbons over here, we must have released some carbon dioxide. I'll make them pink. And I'll put them in a circle. That's usually what textbooks do for whatever reason. Carbon dioxide is always in a circle. We do not produce any ATP at this step, but we do produce more NADH. I think I was using green yesterday. 1 NADH for each of the pyruvates that gets converted. So I'm going to again put 2 NADH plus 2 H plus. I'll put that in a square. Help us find them later. Okay. What's the last thing? Where does this happen in the cell? Because we're in eukaryotes, this actually happens in the outer membrane of the mitochondria. So that we learned all the parts of the mitochondria in chapter 6 was super important because now we're going to use all of those parts of the membrane. So I'm going to share my screen with you because now we can take a little rest. This is stuff that you had to write down. Now I'm going to take a little rest and look at some pictures from our textbook. So I'll put this up. So here is the exact chemical reaction. Of course, the enzyme is not named, but you can see it. It's a purple molecule here. It happens to both be a transport molecule and an enzyme. Here is the chemical structure of pyruvate. Do I expect you to remember this? Am I going to have a picture of this and say, what molecule is this? No. What you need to remember about the pyruvate is that it has these 3 carbons. Okay. The conversion, single step, 1 enzyme to produce acetyl coenzyme A here. Notice there's only 2 carbons in the acetyl CoA group. Coenzyme A is joining, carbon dioxide is being released, NADH is being produced. Now, remember again, they're only showing you 1 pyruvate being processed here. We have 2 of them because at the end of glycolysis, we had 2 molecules of pyruvate. So we have to go through this reaction 2 times. 1, 2. Okay. Now you can see they're trying to show you here, this is the cytoplasm, cytosol, this is the mitochondria. They're trying to show you here that's happening in the outer membrane of the mitochondria. I'm going to scroll back, I think earlier. Yeah, this was pretty good. So glycolysis out in the cytosol or the cytoplasm, and the oxidation of pyruvate or the prep step happens here in the outer membrane of the mitochondria. Then we're going to see the citric acid cycle is going to happen in the matrix of the mitochondria, which is that fluid-filled space between the folds of the inner membrane. And then the very last step, step 4, the electron transport chain, that is actually going to happen inside the inner membrane. Those proteins are all going to be lined up in here in the inner membrane of the mitochondria. Okay, so glycolysis, prep step, did all the parts, starting molecule, ending molecule. What did we produce? 2 carbon dioxide, 2 NADH, plus 2 protons. Where does it happen in the cell? Outer membrane of the mitochondria. Yes, and you will have to know that level of detail. You will have to know all of those things. You'll have to be able to remember them. Okay, now the next step, step 3, is a little more complicated. Step 3 is the citric acid cycle. See if I can find a picture that I kind of like here. I think I'll use that one. I'm going to take this down for a second and I'm going to redraw this picture on my little chalkboard with a lot less detail. But I want you to notice a couple things and then we'll come back to this. So don't panic, and it's in your book, right? It's figure 9.12. There are 8 enzymatic steps, right? So all of these white lines here represent an enzyme. They're numbered 1, 2, 3, 4, 5, 6, 7, 8. You don't have to know the names of the enzymes, clearly. These are all the intermediates in these white boxes. So what that means is when this enzyme grabs onto the acetyl-CoA and oxaloacetate, it's going to produce the first molecule of the Krebs cycle, or the citric acid cycle that's produced is called citrate. Again, do you have to recognize the structure? Do you have to know how many carbons? No, go to chemistry, go to organic chemistry, go to biochemistry, learn all about it. This is Bio 1. So I'm going to put up on my board what I expect you to remember from the Krebs cycle or the citric acid cycle. And then again, we'll come back to this picture and look at it again in a little more detail. So I'm going to stop that share and I'm going to erase my board, and then we're going to talk about step 3 here. Okay, so step 3 goes by many names. Your textbook is referring to it as the citric acid cycle. All of the names are going to have cycle in them because it's a circle, okay? Meaning there's really no beginning or end. It's running all the time. It's called the citric acid cycle because citrate or citric acid is the first intermediate on the cycle. A more old-fashioned name, but a name that some of your chemistry books or your anatomy and physiology books or your microbiology book may use, it's also known as the Krebs cycle. With a K, K-R-E-B''S. Krebs is the guy who figured out the intermediates, named some of the enzymes. Originally they had different names, now they are all named for the product that they make. However, apparently Krebs took all the credit, even though the people in his lab did most of the work. So it's not in fashion anymore as much to call it the Krebs cycle, because although he got the money, which is what primary investigators do, he didn't give anybody the credit, any of the credit they deserved for doing all of the work on the intermediates. He just took all the credits and put only his name on the papers. So this is kind of in or out of fashion, depending on the age of your professor and what textbook you're reading. And the third name for this very same cycle is called the TCA cycle. And students often think that stands for the citric acid cycle, but it doesn't. That's not what that is for. TCA, this is the more chemistry name, more chemistry version. It stands for the tricarboxylic acid cycle. Okay. I will probably use all 3 of these intermittently, again, on purpose, not to confuse you, but to get you familiar with all 3 names, because in other classes, someone may call it something else. And I don't want you to be sitting there next semester going, I don't know what this person's talking about. It looks kind of like the citric acid cycle, but is it the same? It is the same. It's all the same thing. So I usually draw the cycle like this. Over here to the right, I think it's important that you make a little chart. And I always write above that, I write 1 turn and then I write 2 turns. Okay. Because remember, we have pyruvic acid. Every textbook in the world is only going to show you 1 pyruvic acid going around the Krebs cycle. We have 2 of them because we started with glucose. Okay. So you're going to need to know the numbers for a single turn of the Krebs cycle, as well as if we are oxidizing glucose all the way, we have to go around twice, just like we had to take each pyruvate and turn it into an acetyl-CoA. We have to take each acetyl-CoA and turn it around the Krebs cycle. Okay, so I'm going to put the acetyl coenzyme A here, like this, coming in with, I believe it's oxaloacetate. And again, like I said, the first intermediate is citrate or citric acid. And then we go around. The rest of the names of the molecules are not important to me, okay? The only reason I do this is because most of the time students get very hung up on, I said we have to know these three things, the starting molecule and the ending molecule, and they want a starting molecule for the citric acid cycle or the Krebs cycle. So I tell them, okay, the starting molecule is acetyl-CoA plus oxaloacetate to give us citrate. Okay, so it's like 3 starting molecules. All right, I need to cheat. I think it's here. We produce an NADH. The very next step, we produce another NADH. At the very bottom of the cycle, we produce an ATP. Those of you that have already had chemistry, you know it's not really ATP, it's GTP that gets converted to ATP. By one, we're just going to do ATP. We make an ATP in the happy yellow sun. Over here, we make a new molecule that we haven't seen before called FADH2. I like to put that one kind of in a triangle. You can think of it as a cousin to NADH. It has a flavin molecule instead of nicotinamide, but it's also adenine dinucleotide. We produce another NADH up here right before the end. And we release some carbon dioxides, which I don't know, CO2 here in a circle, and CO2 here in a circle. Okay, so on my table here, I would make a list and I would put the molecules that I'm trying to follow down the middle like this. I usually do NADH, CO2, because those come first, ATP, and FADH2, like that. If I go around the Krebs cycle one turn, I'm going to produce how many NADHs? Anybody? Somebody? Anybody? 2, 3, you're right. 3, sorry, my fault. Yep, I'm going to produce 3 if I go around one turn. How many carbon dioxides am I going to make? 2, 1 ATP, and 1 FADH2. If I go around twice, I'm going to double all those. Okay. And remember, why do I have to go around twice? Because I started with glucose. And quite honestly, guys, usually right above this, I write, if I started with glucose, then I must go to Krebs. But clearly I don't have the space. So I usually write that. And the reason is because this cycle, which we should probably put here in the middle, this cycle is used by cells anytime they produce an acetyl-CoA. And I'll tell you right now, you can digest a fat, you beta oxidation of fat to produce acetyl-coenzyme. If you digest proteins, at times you're going to produce acetyl-CoA or some of these other intermediates. So this cycle is central to all of metabolism, pretty much in all living things, because remember, plants, even though they can take light energy from the sun and turn it into ATP, they need to then produce glucose, and then in the parts of the plant where they don't have chloroplasts to be able to make more ATP, they actually break down glucose into mitochondria, and they use the citric acid cycle. Protists use the citric acid cycle. Bacteria use the citric acid cycle. Viruses, no, not living things. They don't have this. Right? Where does this happen in the cell? Matrix of the mitochondria. So if we go back and take a little bit of a rest here and take a look at some pictures, right? So you can catch up on your writing. Here again is the citric acid cycle in a lot more detail than I've given you. I expect you to know it the way that I put it on the board. Those are the minimum amounts of things that you need to know. Again, they're showing you all the names of the intermediates, don't care. They're showing you here where the carbons are actually coming from, when they are pulled off of this molecule, and which hydrogens are being added to the water molecules. Again, don't care. Where does this happen in the cell? Let's go back to this picture. Right, citric acid cycle matrix of the mitochondria. In this, it's actually in this lighter colored yellow-orange spaces here, this fluid. This is the place where the ribosomes are for the mitochondria and where the single chromosome for the mitochondria would be located. Okay. I don't know, I guess they're just showing you step by step, whatever. Okay, now the last step is the hardest. Okay, the last step is the electron transport chain, step 4. This was step 3. Step 4 is called the electron transport chain and ATP synthase. Some textbooks, you'll also see it called chemiosmotic coupling, right? I'm probably not going to use that word very often. It'll probably show up more in the homework than it will on the quiz. Because again, it's Bio 1. I like to try to keep it as simple as possible. You can add more layers of complexity on as you further your studies in biology and chemistry. So I'm going to erase this because I think I'm going to draw the electron transport chain, like a poor version of the electron transport chain on the chalkboard here, and then we can go through it. I know that in your note outline, all of the words are written out, and I'm going to go in the same order, or try to go in the same order. If I forget anything, give me a hand signal in the chat so that I can be sure that we're complete. All right. So, behavior axis switches. Very last, number 4, very last part is we call the electron transport chain. That's what the ETC stands for, electron transport chain, plus an enzyme, ATP synthase. Right? And the enzyme, ASE, that's how we know it's an enzyme, and it tells us what it does. It synthesizes ATP. It makes ATP. So we're going to talk a little bit of how that happens. So we're going to start with a membrane, because the electron transport chain, depending on what kind of organism you're in, there are some variations. The electron transport chain is at least 9 proteins in a membrane plus ATP synthase. Okay, so I'm going to draw, if this was the mitochondria, here like this, right? And this is the outer membrane. And this is the inner membrane folded up. Right. I would remind you that, so this is the matrix then in between the spaces here, the part that I'm kind of shading in, right? So this is the outer membrane, this is the inner membrane. Always, all membranes in all cells are made of a phospholipid bilayer, right? So if I were to just make my membrane here, it's actually going to be from the inner membrane, like a blow up right there, folds the cristae, the inner membrane of the mitochondria, it would look like this. So I would have my phospholipid bilayer, which I'm sure you recognize from chapter 5 and chapter 7. So put a few phospholipids there, and then I'm going to get out my purple chalk and I'm going to draw some proteins in the membrane. similar to the picture in your book. There's one down here, and then there's another big protein here, and then there's one up top, and then there's another protein here that kind of looks like a tooth. Right, like that. And then there is ATP synthase, which kind of looks, it's a two-parter, kind of looks like this, and it has a round portion here at the bottom. I usually kind of draw it kind of square, and then some connector molecule between them like this. That's the ATP synthase. I'm going to fill in the phospholipids in between real quickly here. So I'm taking the time to draw this rather than just flash a picture at you from the textbook, because if you can draw this, and if you draw it and you think about it while you're drawing it, it will help you remember the electron transport chain and the parts. You'll remember it much better if you actively do something rather than just click some buttons or look at a picture or watch a video. Right? So that's why I'm doing this, because I'm hopeful that you're going to do this at home. Right? Crazy? Okay, so I know you can't see the color very well, but these are purple. They are proteins in a membrane. And you might be looking at me and you might be thinking, she said 9 or more proteins in a membrane. She only drew 1, 2, 3, 4, 5. She only drew 6 of them. That's because some of these are complexes. Some of them are more than one protein grouped together. Okay? There is a picture of that also in your textbook, which I'm not going to try to draw. It's on the page before. It's figure 913. Okay, so now we can talk a little bit more about electron transport. It is what it says it is. It can transport electrons. And we're going to use a proton gradient in order to produce ATP. Now, how does that work? Okay, so I'm going to draw my NADH friend here from the matrix of the mitochondria. So NADH, right, plus its proton, the reduced form. This is what comes to the electron transport. Where is it coming from? It is collected up from glycolysis, the prep step, and the citric acid cycle. So those 6 NADHs we made from the citric acid cycle, and the 2 we made from the prep step, and the 2 we made from glycolysis. All of those are going to be delivered here from the matrix, right? Because that's the matrix side is over here. This is the matrix is actually on both sides of this membrane, right? Because we're just looking at this inner membrane here and just a piece of the inner membrane. So the NADH is floating around in there because that's where the citric acid cycle is. Citric acid cycles here, 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 multiple citric acid cycles. The NADH comes to the first protein of the electron transport chain. It is going to release a pair of electrons. I'm going to draw them in yellow, and maybe I should do orange. The electrons are going to be passed from one of these proteins to the next. The electrons are passed along the electron transport chain from one protein to the next. So all of these 9 or more proteins in the electron transport chain, they all pass electrons. The final electron acceptor is oxygen. It's going to gain those extra electrons. This is hugely important. And probably on the handout, I probably actually wrote the electron transport chain is 9 or more proteins in a membrane plus oxygen, because oxygen is the final electron acceptor. Burn that into your brain, same as all membranes and all proteins are all membranes in all cells are made of phospholipid bilayer. Oxygen is the final electron acceptor in cellular respiration, oxidation of glucose, or we would say aerobic respiration. That's what the oxygen is for, aerobic, right? Using oxygen. That's just half of it. So we've passed the electrons. Now, at the same time, some of these proteins, in addition to passing electrons, some of them pump protons. This guy is a proton pump, not Q or this guy, but this one, proton pump. And this last one here, which I know you can't see very well, but it's the same as the picture. Right, this is 915. This is figure 9.15. So proton pump here, proton pump here, proton pump here. Now we have accumulated a lot of protons outside the membrane here on one side of the membrane compared to the other side. These protons are going to travel through ATP synthase, and they provide the fuel to produce the ATP. No protons, no ATP. When one NADH is delivered to the electron transport chain, that is enough pumping of protons and passing of electrons to fuel ATP synthase. I'm getting lost over here in the mix. I'm going to make it look a little more substantial. To fuel ATP synthase to produce 3 ATP. So one NADH produces approximately 3 ATP. And again, that's approximately. It's not actually 3, it's less than 3, but that's too much math to try to calculate. So we're just going to say one NADH is going to produce 3 ATP. 9 or more proteins in the membrane plus oxygen. All the proteins pass electrons. Some of the proteins can also pump protons. The protons are the fuel for the ATP synthase to produce 3 ATP.
I have a question.
If you want to look at the picture, we can look at the picture for from your book. If you want to look at that instead, I think my picture's better. Here we go. There's the whole picture. Okay. So the yellow or orange, we see the electrons. That's what this is. Pass, 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 ending up at oxygen. Protons are being pumped here, here, and here. Protons are the fuel for ATP synthase. As the protons get pumped across ATP synthase or through ATP synthase, that's how the ATP is produced. One NADH gives us 3 ATP. Now, our friend FADH2, also produced in the citric acid cycle, can come to the electron transport chain and drop off electrons and a proton, and the proton can be pumped. But see how FADH2 is coming to this second group of proteins here, not the first group? And because it comes here at the second group and drops off its electrons at Q, essentially, these protons are not pumped when FADH2 comes. So we're short fuel for ATP synthase. So a single FADH2 has lower potential energy and will only produce 2 ATP. I'm going to write that up here before I forget. So one FADH2 is going to produce only 2 ATP. Because FADH2 is going to drop its protons and electrons here, okay, not at the first molecule. So this proton doesn't get pumped, so there's less fuel for ATP synthase, so we can produce fewer ATP molecules at the end. where does this happen in the cell? We've already mentioned several times. Hang on a second, Victoria, as soon as I'm finished, okay? Just let me finish this section so I don't forget to give you all the information. Where does this happen in the cell? Inner membrane of the mitochondria. It's also appropriate for you to say the cristae of the mitochondria because it's in the folds. So if I go back up to this earlier picture where your book had this diagram, which is where it is, right? They showed you this in the opening parts of the chapter because they're trying to show you overall what we're going to study in this chapter: glycolysis, prep step, citric acid, oxidative phosphorylation, right? Or the electron transport chain, and chemiosmosis, inner membrane of the mitochondria, the folds. Okay, what's your question, Victoria?
Where it says 1 NADH.
Oh, wait, I can't hear you. Sorry.
Sorry. Where it says 1 NADH, does that say +3 ATP or an arrow?
Oh, no, an arrow. Sorry,
Okay.
an arrow.
Yeah, okay.
Right? So 1 NADH, when 1 NADH comes to the electron transport chain, it is enough energy. It transfers enough electrical energy and enough of these protons to produce 3 ATP. Okay. 1 FADH2 only produces enough potential energy to give you 2 ATP.
And that's coming out of the last, like, little tooth shape?
Yes, so that these NADHs and the FADH2s, they're coming from the citric acid cycle, the prep step, and glycolysis.
Okay, so everything before the ATP synthase. Yep. Okay, yep. Got it. Thank you.
Yep. Okay, where does this happen in the cell? We did that. All right, I'm going to give you guys a second to process this a little bit. And I'm going to see if your book has a picture of, like a summary picture, if I have to write it out. I think there's a table on the handout, isn't there? That has like a total of 38 ATP and where it all comes from, I think. Yeah, so see how in your textbook it always says maximum ATP per glucose about 30 or 32, okay? The reason that they say that is because in eukaryotic cells, you need to expend some ATP moving these molecules around by active transport, especially out of the mitochondria at the end. For our purposes, for our testing purposes, we're going to account for all 38 ATP. We're going to go through and show how we get all 38 ATP, and that's the magic number for us, 38. When you go to anatomy and physiology and they teach you 34 or 32 or 30, then learn that number there, right? But for our purposes, we're going to go through how we get all 38. So I think I'm going to stop sharing this and I'll probably put it on the chalkboard again. Actually, I might do it on a whiteboard. Let's see. Yeah, let's do it on the whiteboard. So in your handout that you have, I believe there is a table that is blank with things for you to fill in for the complete glucose all the way to the end. Count up for all of the energy that can be produced from one molecule of glucose. And I think it looks something like this. Let's see if I can sort of recreate the table. I think it goes glycolysis, and then underneath that it says prep step, and then underneath that it says citric acid cycle, And then next to each of these, there's, or actually it's up above each of these, right? So it would be like this. We could put NADH and then CO2. I can't drop that down, ATP and FA. A little longer here. FADH2. Oops, there, like that. And then underneath there, it would be good if you could fill in the numbers, the correct numbers for each of the steps. So I'm going to give you a minute. And I'll just go through, right? So we made 2 NADH. We made 0 carbon dioxide from just glycolysis. We net 2 ATP. And again, we're going to produce 0 FADH2s. Right. And if I would do the same for going through the prep step, I would have 2 NADH, right? In the prep step, I produced some carbon dioxide, because remember, we're going from 3 carbon molecules to 4 carbon molecules. So we release 2 CO2. In the prep step, I don't think we make any ATP. I should check on that. And we don't make any FADH2s either. I think it's just NADH and carbon dioxide. And then for the citric acid cycle, and we're completely oxidizing glucose here in this example, so I'm going to put the numbers for going around twice. All right. So altogether, from the chart that we had up on the chalkboard, 6 NADH and 4 more carbon dioxide get released. We make 2 ATP directly, and we're going to make 2 FADH2s directly. Now, of course, the last step is the electron transport chain. And the chemiosmotic coupling with the ATP synthase, right? So electron transport chain, I guess I should have put electron transport chain plus oxygen plus, you know, ATP synthase. And then under this, I'm just going to draw, right? So if we look at how many NADHs we have total to bring to the electron transport chain. Right? How many total do we have? 2, 4, 10, right? So let's put 10 here. We have 10 NADH total. There we go. 10 NADH total. So now where are they going to go? They're going to go, and I'm going to make them purple to remind you, right? They're going to go to the electron transport chain, and I'm just going to draw it kind of as little lumps of, to remind you, those are the protons, proteins, right, in the membrane. These NADH are going to go to the electron transport chain, and if I have 10 NADHs at the electron transport chain, how many ATP am I going to produce via ATP synthase here at the end? Draw my little lollipop. Little connector. That's supposed to be ATP synthase. By pumping the protons and passing the electrons, I would make a total, because for each NADH, I produce 3 ATP. So 10 times 3 would be 30 ATP. 30. This yellow is not as bright as I would like, but it is what it is. And I'm going to try to draw my happy yellow sun around there. Right. So just from the NADHs, from collected up from glycolysis, the prep step, and the citric acid cycle, I have 10 NADHs, and that's enough to produce 30 ATP. I still have these 4 ATP that I made directly, which we would call, some books you'll see that called substrate level phosphorylation. So I'm just going to put an arrow here. Yeah, we'll leave it yellow. What the hell? All right, so just from that, I've made 4 ATP. Sorry, I'm kind of making a mess here. My brain's going faster than my hand. No, I just want a line. There we go. So I'm going to make 4 ATP directly. And again, I'm going to put those in another happy yellow sun, which I'm sure that looks just like the sun. And now we still have our friend FADH2s, which I was making orange, so I'll keep that in kind. So that looks too much like the yellow. Let's make them red. So these 2 FADH2s, remember, they also can go to the electron transport chain, right? They don't drop off as much potential energy as the NADH, but they still come to the electron transport chain. And each FADH2 can produce 2 ATP, and I have 2 of them. So 2 times 2 is going to give me 4 more ATP from my FADH2s. 4 more ATP. I'll say that again because FADH2 provides enough potential energy to make 2 ATP from the electron transport chain and chemiosmotic coupling. And I have 2 of them, so 2 times 2 is 4. And when I add these up, 30 plus 4 plus 4, that gives me the big grand total of 38. Total ATP from 1 glucose. Now, this is super if all systems were optimized, which is also part of the reason why your book says it's 30 to 32. Not only some of these are used for transport, but also not all cells can make this maximum amount at all times. Okay, I'm gonna pause there, and I see some people are having some questions, right? Victoria, no. So it's the ATP synthase that produces the ATP. So before the ATP synthase, there are 0, or only the ones that we've produced from glycolysis and the prep step, the glycolysis and the one directly in the Krebs cycle. Those are made by a different mechanism, not ATP synthase. It's called substrate level phosphorylation. And that's a different, we're not talking about that.
I have a question.
Other questions you guys have? I know it's a lot. This last bit is kind of very heavy.
I have a question. If we were to add the ETC step 4 to this chart, would
that's what I was trying to do on the whiteboard.
Yeah. would NADH be 10, even though that started before?
Yes.
They move through
because
the cycle.
because they're collected. They literally are sort of collected up and all shuttled to the mitochondria.
Okay, great.
And remember, this chart that I drew here, this is only for complete oxidation of glucose. So if we were in Chem 2, for example, and we were talking about beta oxidation of fat, I would have a different chart, right, with different steps here. I would still have the prep step probably and the citric acid step, but I would have some other steps in front of this, and they would have different numbers. Right, so this is just for oxidation of glucose, aerobic respiration, aerobic cellular respiration. It's most technically correct, I guess. So I'm gonna stop sharing that. But wait, there's more. Sorry. Bad joke coming. all for the low, low price of Bio 1 at community college. You can also learn about fermentation. So we have to do two more examples. And for that, I'm not going to draw those. I will just show you the ones from your textbook. So when a cell does not have enough oxygen to be the final electron acceptor here at the end of the electron transport chain, and some of those hydrogens that are pumped through, of course, are going to form water molecules. But if there is not enough oxygen here for aerobic respiration, some cells have the ability to produce ATP a different way. It's not as much, but it can be fairly quickly, and that process is called fermentation. I'm going to show you two examples of fermentation: alcohol fermentation and lactic acid fermentation. So this is alcohol fermentation. This is using glucose to produce ethanol. Notice we still have glycolysis, that whole big blue arrow there. All the steps of glycolysis, which is going to produce 2 ATP and give us our 2-pyruvate. But the pyruvate are not going to be converted into acetyl-CoA, and they're not going to go to the citric acid cycle. They're going to go through 2 more enzymatic steps and produce ethanol instead. Notice that the NADH that was generated during glycolysis is going to be used to produce that ethanol. Okay, so we do lose some carbon dioxide. So ethanol only has 2 carbons, whereas we make 2 of them, so we'd have 4 carbons at the end, and glucose, of course, has 6 at the beginning. This process only makes 2 ATP because no electron transport chain, no citric acid cycle. How a cell can sustain itself. So if I'm a yeast cell, for example, I'm a fungus cell, or I'm a bacterial cell, I might be able to get enough ATP by fermentation because it's so many fewer steps. It actually can happen super quickly, much more quickly than going through all of the prep step, the citric acid cycle, and the electron transport chain. And if I'm a single cell, I don't need as much ATP as like a whole organism. This second one is lactic acid fermentation. We show you this because this can happen in your muscle cells. Again, when there isn't enough oxygen at the end of all the electron transport chains in all the mitochondria in all your muscle cells can ferment to get a little bit more ATP. This lactic acid then builds up. That's what you feel as the burn when you're exercising, right? And that's telling you that you're in oxygen debt. Right? Your mitochondria need more oxygen in order to continue with glycolysis and produce the maximum amount of ATP. There are several bacteria that also produce lactic acid or do lactate fermentation, and lactate is their end product. There's also a lot of other organic molecules that are the result of fermentation. Again, go to biochemistry, go to microbiology to learn all about it. You just need to know these two: alcoholic fermentation and lactic acid fermentation. Again, notice with the lactic acid fermentation, lactic acid has 3 carbons, so we don't produce any CO2, whereas with the alcoholic fermentation, you do. And the very last thing that I need to show you is this picture. And this is, again, to remind us that what we've talked about yesterday and today, glycolysis, the prep step, the citric acid cycle, and the electron transport chain and chemiosmotic coupling, that's central to all of metabolism. When we break down a fat, we break it into glycerol, which is a 3-carbon sugar, and it's going to go into glycolysis. And then all of the fatty acids are going to be broken off 2 carbons at a time and converted to acetyl-CoA. This is how fats store so much more energy than carbohydrates. Proteins, notice we can degrade a protein, we remove the amino group, and then there are several places where the rest of the pieces of the amino acids can get put into this central pathway: glycolysis, aerobic respiration, cellular respiration, whatever you want to call it. Likewise, when we have excess, these arrows can all go out the other way, right? Or when the cell needs to build proteins, some of the components of the intermediates on glycolysis, we don't use it to make ATP, but we're going to use it to produce amino acids so we can build proteins. I think that's what the other picture is supposed to be showing us. What's this guy? Yeah, we're not going to talk about that. Okay, that ends what I'm going